everyone. I am Shin and would like to welcome Dr. Jaime Jayapalan. We are from the Department of Molecular Medicine in the Faculty of Medicine, University of Malaya. Dr. Jaime specializes in cancer biomarker discovery and clinical proteomics. Her research focuses on identifying molecular changes, particularly in proteins that can be used for early cancer detection. She is very dedicated to translating these scientific findings into practical, user-friendly diagnostic tools and tests. Let us welcome Dr. Jaime. Over to you.
Thank you, Prof. Fung. You're very kind with the introduction. Okay, without further ado, let me start sharing my screen. Okay, once again, a very good morning to all present, both in the panels and the attendees, all right? So today, I'm actually very excited to share our journey in translating proteins or protein science into simple, accurate tools for early cancer detection, particularly for point of care use, all right? Now, via this seminar also, I hope to inspire collaboration and spark fresh ideas that can help bridge the gap between laboratory research and also patient care. Now, late stage diagnosis, right, leads to poor outcomes. This is something that we know. It is sad, but it is a common occurrence among our cancer patients, including those with prostate and pancreatic cancer. In contrast, if detected early, right, these cancer types, both prostate cancer and pancreatic cancer, has a remarkable 5-year survival rate. Here, as you see, 99% for prostate cancer and 44% for pancreatic cancer. Next, what is biomarkers or what are biomarkers? Biomarkers are measurable indicators found in the human body to indicate whether it is normal or the body has some, has undergoing some abnormal processes, or it can even indicate the presence of disease. Giving you an example, the most simplest biomarker I can think of now is blood pressure, all right? Or we can even use ammonia as biomarkers, to detect the levels of ammonia to indicate whether a person have liver dysfunction, for example. Or we can even, we can always fall on our typical biomarkers that we use in our hospitals like PSA, troponin, I, and T. All right. Now, talking about biomarkers here, let's focus on my cursor here. Okay. Biomarkers or biological markers such as DNA, RNA, metabolites, can be sourced from a variety of bodily fluids and also tissues. Giving you example, the blood, saliva, CSF, the list goes on. Okay. But what's important here is that how these biomarkers play a critical role in diagnosing disease, for example, monitoring disease progression, or to be used as a screening marker, or even for predictive purposes. All right. Now, next is on proteomics, right? Large-scale study of proteins in human disease, in human tissues, cells, or bodily fluid is known as proteomics. Okay, it is a very essential or very wonderful technology to use to understand disease mechanism and underlying potential biomarkers. The most common technique used for proteomics approach is two-dimensional gel electrophoresis. This is a bit outdated though. All right, mass spectrometry, the current core proteomics platform people usually use. And also there is also the other thing called protein microarray. Okay, now talking about clinical proteomics, the application is very diverse. All right, it runs, as you can see here, across board, from identification of biomarkers until the advancement of personalized and precision medicine. Here, I want to highlight our research focuses on identifying reliable biomarkers for early detection of cancer, intending to improve outcomes through timely intervention, right? This slide here highlights our early work, which began like 15 years ago. Yes, we started small using conventional 2D electrophoresis technique and explored a wide range of protein samples, including the neat sample, processed serum, and urine. enriched serum samples, whole saliva, oil palm root proteins, small animal tissues, whole fly proteins, and even hair shaft proteins. Over time, as I've mentioned earlier, as technology advanced, so did we. So now, if you come to our lab, or all our projects have now embraced this evolution by incorporating mass spectrometry and using mass spectrometry as our core analytical platform in all our current studies. So going back, sorry, I just want to, okay, when you look at this slide, right, it may look fragmented at a first glance, but if you put it on a global scale, okay, all the project listed or all the project that we have involved in the past 15 years, they align cohesively and contribute meaningfully towards the overarching goal of personalized medicine. While the individual projects may have varied endpoints, okay, due to differing objective or resources or even circumstances, but one thing is constant is the commitment towards advancing precision medicine throughout the years. All right. So for this seminar, I've chosen to showcase two key projects here, one on prostate cancer and the other one on pancreatic cancer. Now, let's look at prostate cancer. It is one of the top 3 cancers in men. Okay, 9.3% here. And it is often diagnosed late, right? Meaning that the treatment has become more challenging. Okay, the key issue lies with the existing method or existing diagnostic or screening method, which is the PSA, prostate specific antigen. The commonly used prostate-specific antigen threshold is highly debatable, whereby we can say it lacks tumor specificity, meaning that it can go both ways. It can show you false positive or even false negative. Okay, because of this, there is an urgent need for better, accessible diagnostic tool that 1, non-invasive, 2, reliable, and 3, suitable for use even in primary care or community settings. So here we are. So inherent to this demand for better tools, after identifying promising biomarkers in serum and urine, we ask ourselves, how can we turn this into something practical, right? So the answer we got, thank God for good collaboration with Biogen's technology and good funding support from MOSTI. So the answer we got then was biosensor. Biosensor is a tool that is quick, cost-effective, and portable, designed to detect cancer-related proteins accurately even outside of major hospitals. So this would then bring diagnostics closer to the patient, especially in resource-limiting settings. Now, next one is the pancreatic cancer. It is the 12th most common cancer in the world. Okay, and among its variant, the PDAC subtype is the most common ones. Sadly, guys, PDAC is highly fatal, okay, or they're one of the deadliest cancer because it shows very little early symptoms, okay, meaning that by the time it's diagnosed, it is often too advanced for any implementation of effective treatment. All right, that's why this is the main reason why early detection is critical and also extremely difficult with current tools when it comes to PDAC. Let's look at the current diagnostic tools that we are using in the clinic. All right, so the current tool that we are using for pancreatic cancer detection is CA19-9. Okay, although it is the FDA approved biomarker, CA19-9 is actually, it lacks specificity and it is more useful for monitoring disease progression and treatment effectiveness rather than for early diagnosis. Additionally, imaging methods like MRI or CT scan, yes, we can use it, but it is often invasive, expensive, and may miss early stage cancer. So adding to this complexity, PDAC is not a single disease. Unfortunately, it has distinct subtypes, all right? Meaning it has unique genetic and molecular features, right? As you can see in this slide here, meaning that this subtype influence how the cancer behaves and how it responds to treatment differently. So this biological diversity means that there is no one-size-fit-all diagnostic test, as what Dr. Tan Kim Ki said earlier, but for a different context, but it applies here, okay? It's unlikely effective, lah, okay? So we need a more tailored, a more diagnostic approach. To address this complexity, we then explore unique strategy using circadian rhythm. So everybody here knows circadian, what is circadian rhythm, right? But just to reiterate, circadian rhythm is our body's natural 24-hour cycle that regulates many body function, as you can see here, all right? But when disrupted, they have been linked to several diseases, including PDAC. So we thought by studying this rhythm, we may potentially uncover unique protein patterns or markers that may have been missed previously using conventional method. So in line with our hypothesis, thank God for that, our analysis showed, okay, several proteins in PDAC cells following circadian oscillation, which differs from the normal cells. Okay, you can't see here, but I'll show you in the next slide. So it differs, but yes, we were excited when we saw this result, but the question that we asked ourselves then was, can these proteins help in early cancer detection? And ta-da, yes, we found that specific proteins are upregulated in different PDAC subtypes, CAPAN2 and PANC1. For example, kallikrein, mucin, cathepsin, kallikrein 8, were linked with CAPAN2 subtype. Meanwhile, proteins like alpha enolase, vimentin, CD44 antigen were elevated in PANC1 cell. So just for information, CAPAN2 cells represents early stage cancer. PANC1 cell represents the more aggressive and advanced stage cancer, right? So interestingly, another interesting point I can demonstrate to you here is that these markers where I mentioned earlier, they appeared at specific time points, highlighting the importance of timing and also the key role of circadian rhythm in cancer biomarker discovery. So having said that, assigning each of these marker to its circadian time window is the key to accurately detect PDAC and also can be used to understand its subspecific type behavior. Of course, turning research into real-world diagnostic tools is not straightforward. First, we have issues with validation. We need to prove that biomarkers are accurate, reliable in real clinical setting. The next one is even my biggest worry here is the funding, okay? So developing and testing medical devices is not cheap. Aside from this, we also have another hurdle that requires, or we need clear regulatory approval. And most importantly, we need collaboration across disciplines to make it happen. Of course, we have wonderful collaborators now, but the more the merrier, okay? But aside from this, this is my personal thought. One small but meaningful way to translate science into real impact is by empowering the next generation. Okay, so here I want to take this opportunity to express my heartfelt thanks to all my students and research team over the years. What you see or whatever I've presented today in this slide are actually truly a collective effort from these people. Okay, so mentoring young scientists have been one of the most rewarding part of my career. They are without doubt at the very heart of my research journey. So what's next with the project? Sure, we are looking forward towards validating the biosensors in clinical setting. We are also planning for integrating AI to enhance the detection of cancer and actively seeking collaborators to co-develop, test, and scale it up regionally. With this, I have a dream. I envision translated science being used in Malaysian clinics, mobile health units, and even home kits, especially to reach the underdeserved communities. Okay, so far we are fortunate, as I've mentioned earlier, we are very fortunate to have worked with excellent collaborators across disciplines, from clinicians, physicians, cancer experts to tech developers. And this is the network that drives our innovation and impact in our research, right? And I'm also very grateful for the generous funding support that has made this work possible throughout the years. This includes MOSTI, MOHE, UMSC CAF Fund, and yeah, and everything, right? Thank you for listening, right? Oh yeah, so this is my slide. I'm actively looking, sorry, a bit promo here. I'm actively looking for collaborators to work with me on validating some markers on a breast cancer project. So if you are very passionate about diagnostics, improving diagnostic for patients and healthcare, please reach out to me. All right, thank you.
Thank you, Jaime, for the interesting talk. Are there any questions from our participants? Okay, I have a question for you, Jaime.
Yes.
I know you're working on proteomics, looking into proteins as biomarkers. Would you also envision that one day we may also look into metabolomics, the metabolites found in our tissues or blood or other human fluids to detect diseases?
Sure. That's another aspect that I'm currently working on, Prof. Fung, metabolomics. It's a study of metabolites, right? So I'm slowly penetrating the other omics aspect, in our current research. Yes, another way forward, aside from protein, you see, we have to answer this question, we have to go back to our central dogma of molecular biology. You see, DNA is transcribed into mRNA, mRNA is translated into proteins, right? But proteins have to be degraded, right? When it degrades, it becomes metabolites, right? So this is another aspect we can, it's a very enriching aspect that we can really investigate to find interesting markers to answer, to differentiate between the healthy and disease state. Now, but when it comes to metabolomics, right, there are a lot of other challenges, that we may have to tackle, because even diet also contributes to the metabolome in the human body, right? So these are the initial project design kind of challenge that we have to tackle first before we can actually go full swing before incorporating that into our approaches. Yeah, that's what I think.
Yes, that's excellent. That's why I think our department is doing possibly an all-rounder kind of research work where some of us look into food, some of us look into detection of diseases. Well, since we are the Department of Molecular Medicine, I think that's apt. So thank you, Jaime, for
Thank
a
you.
very enlightening talk. I would like to conclude today's session and thank you very much for your attention.
Thank you.